since it was in the email I sent out. But it's surprising what you learn about people by just fishing around on the web a little bit. And along with being an expert in genetic genealogy, David Weiderberg is also an accomplished cabinet maker, bachelor's degree in cultural history, involved in genealogy for, I guess, 30 years now.
Yeah.
He's connected with the Norgesprosjektet-DNA, which is part of the family tree DNA. And I think he'll encourage any of us who haven't to do the big Y analysis. We heard from him last a couple of years ago, speaking about genetic genealogy more generally. Tonight or today, we'll talk specifically about Y chromosome analysis and how that can help with tracking some of our ancestors. David, let me make you a co-host.
Yeah. Oh, it's a little. Yeah. Mm-hmm.
Yeah, we see your screen.
Yeah. I'm just trying to wait a little. How is it? Okay. Maybe it's just show it as it is. I'm trying to make it full screen, but sorry. Yeah. Yeah. Yeah, I'm going to talk a little bit about YDNA and how you can trace and confirm lineages. First, for some basics. Everyone sees the whole screen or not?
Yes.
Yeah. Okay. You have different types of DNA. You have YDNA, which is the topic of this presentation, which is inherited only by males. And you have autosomal DNA, which is DNA that you can use to trace the DNA you have inherited from all your grandparents and up to second great grandparents. And you have also mitochondrial DNA, which is used to trace maternal lineages through mothers. And you have xDNA, which is kind of a special type of DNA, which is more advanced level to use. Autosomal DNA has some limits, which is that they can only be used for certainty 5 to 8 generations, and they are sold by most of the other companies. But those types of tests that we are going to talk about are lineage tests, which usually is recommended at family tree DNA. Some of you might have tests at 23andMe. They can also show haplogroups like YDNA and mtDNA, but they don't have any matching material. Yeah. YDNA is found in the Y chromosome, which are what defines you as male. It is only inherited through males, from father to sons, and they are inherited almost unchanged. But at around 3 generations or so, some changes might happen. The probability of a mutation is a little bit higher when father is older. And that is also why we can put these mutations and describe it like a tree, like this. These mutations happens when cells are divided to create sperm. A mutation happens when the genetic code of father is not copied exactly. So in this case, you might have a mutation called N3, like this, and you have inherited all the mutations. You have inherited all the mutations from your father, and you might also have more mutations that your father doesn't have. And with the help of these mutations, you can identify who is your relatives. So you share the mutation M3 with your father and his brother, but you might not share it with your cousin, and therefore we can try to find out which of these ancestors these mutations have occurred in. When you match with unknown relatives, they might have some other mutations like this, but they might share one mutation. And these mutations are what you could use to find out how far back in time you are related. So the number of mutations here that you don't share are what we use to calculate how many generations far back you might be related. So research shows that under 2% of every born child has wrong father. Some might say that that number is very low, but when you divide it by generations, it becomes much more. In YDNA, you can divide it in different periods like this. The bottom area is genealogy that you can confirm by written sources. The next one is the type of lineages that you can confirm by using SNPs and STRs. And the second one is what we call deep ancestry. And that is ancestry back to the Stone Age and stuff like that, and back to our common origin. By the use of these mutations, we can divide the paternal lineages in different groups or haplogroups, like this. These haplogroups can be used to map lineages throughout Europe and see how the frequencies is in different countries. So this is the most common paternal haplogroup in Europe, R1b. And this is R1a. And this is the most common haplogroup in Norway. So you'll see that in Norway, some areas in Norway, almost 50% of all paternal lineages has a common ancestor back to a haplogroup. This is how a match page looks like. This is my father. These are based on STRs. So here you have 3 steps. That means that 3 of the STR values are different from my father to myself. And these others have more differences. So they are likely more distant. And then you have these. These are called, this first one is your haplogroup. And the next one here is the SNP that defines the haplogroup. And at last here, you have a name of the earliest known ancestor of the tester. That is very important information. So Joel, you see here, I have a Myklebust myself on my paternal lineage here. But it's not the same Myklebust. It's a different one.
I guess there are a lot of them.
Yeah, there is. Yeah. Okay. This is how results are presented in our project. All these values are STR values. Don't know if anyone understands this kind of a page. Yeah, but that's why we're that's why you should join that project, because all of these groupings are done manually by me and the other admins. And then we also try to put you in different groups like this. This means this is your haplogroup E, and these ones are the mutations or SNPs that you're probably or are positive for. So the 2 different kind of mutations that are important to know about when you're talking about YDNA. This is how our DNA looks like. And here, a SNP is the mutations that occur on one location in our YDNA, while STRs are patterns of repeating codes like this. These ones. So when you're looking at this table, you're looking at STR markers. And these ones are SNPs. Family Tree DNA has also made a new project called Discover. And here I can calculate how far back my lineage is related to a lineage in Sunnfjord, in another county. So they are related around the year 1400. And then I can go further back, and around the year 600 to 700 or so, I'm related to a Scottish branch with the clans MacQueen and with little Mac Isaac. To me, this is fascinating because the origin story of the MacQueen clan is that they are descendants of a Norwegian Viking called Svein. So by the use of my DNA, I can confirm that their story is likely true. Here I have put all the Norwegian lineages in one tree. This is my test, and I have tested one other lineage sharing an ancestor born 1691. So I've confirmed that my lineage is correct back to this one. With written sources, I know that I descend from this one, Olav Eriksson, who lived on Myklebust. But I have also have a match with some other lineages in the nearby valleys. And then I can use the SNPs or the mutations to calculate how far back all these lineages are related. So here you have some lineages in the same county, and here is some lineages in the neighboring county, and all of these should be related around the year 1400. So then I can make also a map like this. Here is my lineage, and I can go further back to the year 400. And then I can connect to an archaeological sample that was found in Greenland, a Viking settlement. And this one is related to a lineage from Gudbrandsdalen in Oppland. So here I have a lot of lineages that I can confirm have a shared ancestor. Family tree DNA has also made it possible to see how far back you're related to archaeological samples. So they have tested skeletons from 3 different sites in Norway. These are the oldest samples in Norway. 7,502 before Christ. That is a long time ago. Before our time, I think it says. Sorry. And you can also have, you can also link to notable connections like this. Franklin Roosevelt, that everyone has heard of, I think, even in Norway. So he shares also ancestor with me, but it's a little bit far back in time. Yeah, and here are a little list of of some tests that have been done. Hans Nielsen Hauge is probably known for some of you, and Ivar Aasen, and yeah, the last kings of Norway before we came under Denmark. We have their YDNA. So by the use of YDNA, you could maybe find out that you are descendants of some royals or maybe some of these famous people. And the current king of Norway, we have his YDNA as well. It's shared with the Tsars of Russia, Romanov, and you even have Thor Heyerdahl. This is probably a known family for some of you, the Gautam family, which is a noble family of Norway, one of the last ones that are still existing in Norway. We tested one lineage that today is called Gautam. We tested their YDNA, and their earliest known ancestor is Johans på Aga. His origin is totally unknown. We don't know who he really was. But to confirm that this lineage is correct, we did find a descendant of this one. And we tested this one as well, and everything matched. So we know that these two lineages are correct all the way back to the 1400s. And to me, that is one of the powers of YDNA, is that they can be used to confirm these long lineages. Because these are a lot of generations with faithful mothers and fathers. Yeah, and this is probably one of the families that Joel are researching. Is that correct?
Yeah, it looks kind of familiar.
Yeah. Yeah, and we have also tried to find out some of this, because a lot of the genealogies are maybe not guesswork, but probably not completely correct, because we have tested descendants of two that should have been brothers, but they don't match each other, these two. But this one had a match with one other one that lived on the same farm, descendants of this one. So we know that these two had a shared ancestor. And this shared ancestor is calculated to have lived around the year 1500. But what about this one? He also had a match, but none of these. He had a match with this one, who also lived on Annes, where he also lived. So these two are probably related around the year 1450. And that is a little bit further back. But the next step in this one is to find out which of these are actually the son of this one, Anders Mattsson, who lived on Annes. To find out which one of these are descendants, we need to find out if he had any siblings that have descendants today, or maybe this one. That's the only way to find out which one of these are really his sons. This family is probably also known for some of you, the Skank family. The Skank family has a lot of genealogies, but a lot of them are not very reputable. And the reason is because a lot of people think that surnames are only inherited paternally. And a a lot of people, both in Norway and in America, think so as well. But we know that in this family, the Skank surname is probably something that they like to inherit or keep. So a lot of these connections that we thought share the paternal lineage here are probably through their mother instead. And what they also have done in these genealogies is to connect everyone which had a coat of arms or a bumerke with a leg in their shield. So every family with something that looked like a foot was connected into their genealogy. And we have then tested around 7 different lineages where none of them share an ancestor at all, paternal ancestor. But some of these lineages do share a common ancestor. So we know that the Skank family has a known ancestor, Peder Jensson Skanke, born around 1570, that we know are correct. But we have a large family in Røros, Skanke, that we now know that are not the descendants of Peder Jensson paternally. This one is a family up in Hallingdal, which is pretty famous for genealogists up there. We had the three that we thought were brothers, who was mentioned in a probate: Bjørn, Kjetil, and Gjermund. And then we have tested descendants of these two, but these two does not match each other. So they are probably not brothers. But we also tested the family on Gulsvik, because genealogists have also had a theory that these also shared an ancestor with this one, Guttorm. We tested this family and they did not match any of these ones. But all of these shared an ancestor born 1484. And to confirm lineages going back to 1484 by both written sources and DNA, that is pretty good. Oh, sorry. Yeah. Okay, I think I'm going to show maybe a little bit how the interface looks like in family tree DNA. But before I do that, I wanted to let you ask some questions, maybe. Okay. Do you have any questions, Joel?
Yeah, I see the Skanka family.
Yeah.
My connection to that is, I think, through Christopher Transon Rustung.
Yeah. Yeah, he's pretty famous too.
Yeah. Does he show up in there somehow?
No, he's not paternally the same family.
Yeah.
Yeah.
David, I have a question.
Yeah.
When you show the family tree information, are you going to describe actually what the project is? Because I'm not familiar with it, but I have some data on the family tree.
Yeah. Yeah,
Oh,
I can show a little of it.
Oh, that would be great. Thank you so much.
Yeah. Mm-hmm.
I have a question also, and that is, I'm just getting into DNA research. And so if I had a grandmother who came from Norway, it's a
Yeah.
grandmother with no known siblings. So
Mhm.
is that line done and finished as far as being able to track it?
Could you repeat it again? Sorry.
That's okay. My grandmother came from Norway
Yeah.
and she has no known brothers or sisters.
Yeah.
And so is my tracking via DNA finished with the YDNA?
Yeah. YDNA can only be traced through males.
Yeah.
Yeah.
Okay.
But autosomally, it should be possible to trace it. Yeah. Because autosomal is it's not that easy to use far back in time, but around 5 to 8 generations is probably the limit for how far back.
Okay, thank you.
Yeah.
Yeah, this is
Yeah.
really fascinating stuff.
Yeah. This is how the interface at the family tree DNA looks like. So these are my YDNA matches.
I think we're still seeing your PowerPoint screen.
Oh, sorry. Sorry. So, stop here. Here. Okay. This is the start page of my family tree DNA, which is my YDNA results are here, autosomal DNA and mtDNA. So this is how my matching page looks like on YDNA. This is my relative that I share an ancestor born 1691 with, and I can use some tools to understand how far back we are. Should be sorry. There. These are based on the STRs, and then I have a date around 1650 with it, which is pretty good when I know that we shared an ancestor born 1691. So here are some statistics for those who likes numbers and mathematics. Yeah, and then they have these different types of reports. This is the tree that I showed in the presentation. So I know that all these should share an ancestor born around 1400. But I know that one of these shares an ancestor born much much closer to our time. So then I use STR in addition.
Yeah, looking at your the spreadsheet that you have with the STRs and so forth.
Yeah.
This is sort of a basic question, but they're all just numbers. What do those numbers mean under each?
Yeah, these numbers. I can show you a little bit of that. If I find the project here, Norway. There. These. You meant like this?
Yes.
Yeah, yeah. If I go down to myself, this is mine. The number here, 13. It means the number of times the STR marker this 393 has been multiplied in your YDNA. So yeah, it's a code. Like it says ACT, for example, and it can be multiplied several times in your YDNA. So if ACT is multiplied 13 times, it just says ACT, 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 ACT in your YDNA.
So what you're looking for connecting between people is how similar these numbers are.
Yeah, yeah. So if I go, here is my test, and these numbers are almost the same all the way, but sometimes one of these numbers will change.
Okay.
Yeah. Like, this one. Here you have one that has changed, and everyone else is the same. Then this one has a new STR mutation that the others doesn't have. Yeah, and what I do as an admin is that I look at all these numbers and I identify patterns. Like this one, this pattern 12, 14, 16, 16 is a signature pattern for my branch. So you have all these ones have the same pattern, 12, 14, 16, 16. And then you have others that have a different type of a signature, like this one.
Interesting.
Yeah. So that is STR markers. But sometimes you have STR markers that changes when you have this one, red. Red means that it's a fast mutation. It changes often, while these blue ones, they don't change very often. So the mutation rate of all of these are different. Does that answer your question?
Yes.
Yeah.
I've noticed in going to the website that periodically the what do you call it? The last SNP, the terminal SNP changes. So you're figuring out new SNPs to test for.
Yeah. I can also show you what that would look like. And maybe there. Yeah, this is a haplotree, and these are SNPs or mutations. You see that here there's just one mutation that are shared by all of these. And up here you have more mutations that are shared, 4 mutations. All of these 4 mutations are shared with this branch and this branch.
Yeah.
And if I have one more tester who gets this one, Y22010, but does not share 2 of these ones, then we get a new split in this tree. Yeah. What?
I finally figured out from staring at this long enough that FT means it's a family tree DNA step that developed.
Yeah. So these are SNPs and not STRs.
Yeah.
So there's 2 different types of mutations. As I mentioned earlier, SNPs, they are mutations that occurs in only one position in your YDNA. Yeah, so they are kind of very specific mutations.
David, how many people are participating in the Nordic DNA project? Because you're dependent on people, agreeing to tests, submitting their data.
Yeah. In our project, there's around 10,000 tests registered.
Really?
Yeah. Yeah. So there's a lot of tests, but not everyone is a YDNA test like this.
Oh, so what kind is that?
Most of the tests that are done are autosomal tests.
Oh, I see. Yes.
Yeah. And that is tests that you can get at MyHeritage or Ancestry or 23andMe as well. So if you have a test at Ancestry or any of the others, you can transfer it to the Norway DNA project and also to family tree DNA.
How do you do that?
How do you do it?
Yeah.
Yeah, it's also show that. Maybe I have to not be logged in, maybe. Sorry. Here, upload DNA data.
Oh, okay. And if it's already in family tree DNA, want to join a group project, my menu options across the top would look like that, and I click Norway.
Yeah, I could show that as well.
That'd be great.
There. Okay. Here it says join a project.
Ah, okay. Group project.
Join a project. And then I can, some of the projects have entered surnames or keywords. It should really be keywords. So if I put in Norway here, it should be possible to find some here. Norway DNA.
Gotcha. Okay.
Yeah.
Thank you.
So it should be possible to find it. So we have 10,022 kits registered in our project as of date.
And what is the one immediately below that? Norway-MRO. Oh,
yeah, that's
county. That's just
that is our county projects. So every county in Norway has their own DNA project.
Oh, really?
Yeah.
Oh,
So if your lineage is from Rogaland, you join the Norway project
right.
with the regional project of
Gotcha.
Rogaland.
Okay, thank you.
Yeah. But of course also the main project Norway.
Hmm.
Yeah. Yeah. Did you have more questions?
There's
have-
one in the chat room.
Yeah. Yeah. Wait a little.
Says, how are you able to get DNA 4 to 5 generations? Skeletal remains?
Yeah, that's a good question. Yeah. Wait a little there and here. Yeah. If you see this this tree are connected to people that lives today. So by comparing these ones and these ones, I have a couple of SNPs or mutations that they share. And these SNPs, they are from a common ancestor. Because when one mutations happens in one man who lived 600 years ago, or maybe 400, all his descendants will inherit that specific SNP. But what you can also do is to compare an archaeological sample with this as well. And what you also use then is SNPs or STRs.
Yeah, I guess Viking DNA is kind of a hot topic these days.
Yeah, it is. Yeah, there's a lot of big studies on Viking DNA. So yeah, it's a lot of lots that you can find. So if I go to, is it this one maybe? Yeah, this one. If you see here, this is an archaeological sample that we know archaeologically is from the Viking era. And I share some SNPs or mutations with him that can be traced back to a shared ancestor who lived around the year 100. But it can also be a little closer than that.
Interesting.
So, yeah, but I don't need DNA from an archaeological sample to confirm if two lineages share an ancestor, because both of those that are tested has YDNA in them that I can use to analyze how far back they are related. Like this.
Yeah.
Yeah, these are a list of SNPs that are shared between these ones and these ones. Everyone here and everyone here has all these mutations. So I don't need the ancestor up here to confirm that this lineage and this lineage shares an ancestor. I don't know if that answers the question in the chat, but maybe.
But if you were trying to find, say, the 6th or 7th generation ancestor, even
Yeah.
if you could match several people across, you
Yeah.
still need to do a paper trail to get the names of those people. Is that correct?
Absolutely. Yeah. The
Okay, so
DNA
it's a
can't,
combination.
yeah, it's always a combination.
Okay.
So when if I go back to the
Yeah, David, one question I
Yeah.
forgot to ask you. We're recording, but
Yeah.
are you willing to share your PowerPoint with us?
yeah, absolutely.
Thank you.
Yeah, it's like this one.
Okay.
Yeah, because I don't know for sure if this one is the ancestor of these two. I only know that he's my ancestor.
And you know that not only from DNA, but because you've been able to do a paper trail to get the names.
Yeah, I have a paper trail back to this one, 1691.
Okay.
Yeah, this is a paper trail and confirmed by YDNA.
Okay.
And with the paper trail, I can also confirm it back to Olav Eriksson, who was mentioned in 1574. And I have done the same with this lineage. I have confirmed it both by paper trail and YDNA back to 1642. And then back to this one, mentioned around 1570 to 1605. But I don't know for sure the connection between these two. That can also be only be confirmed by written sources, but there's probably no sources that far back in time.
But the bottom three folks, for example, what the
Yeah.
three clustered,
Yeah.
they share DNA in part with going back. So it's all the way back to Mr. Ola.
Yeah. I can't know that can't know for sure genetically that everything is correct further back than this one.
Okay.
But I can know genealogy. The genealogy is correct back to this one.
Okay.
I can't know for sure that all the generations up here were faithful.
Sure.
Could have been
Exactly.
something wrong here.
Exactly. Yeah.
Yeah. So this lineage and this lineage could have been related much closer, but I don't know for sure.
Yeah. Visiting farmer that does it
Yeah.
every time.
Like that. It's just a fjord between this lineage and this lineage up in Møre and Romsdal. So it's a maybe 1 hour with swimming. So
Exactly. Sure.
Is there any grave robbing going on?
maybe I'm not sure. Yeah, but the DNA that these have and these have shows that they should be related around the year 1500. So when my ancestor is mentioned in 1574, I think I'm pretty close to a connection here. Yeah.
We have another question in the chat room from Cynthia. She
Yeah.
wants to know if it's possible to still join the YDNA Norway project if just one of your parents is Norwegian.
Yeah, the Norway project is for everyone that are descendants of Norwegians. So it doesn't matter if it's paternal or what it is. So she's welcome. Absolutely. But the result lists in the project are they are restricted to Norwegian lineages. there. I go back to these lists like this. They're only Norwegian lineages. And by that we mean the lineages that we know that have Norwegian origin genealogy back to maybe around 1500 or earlier. So we usually don't include those that have a known Norwegian ancestor born 1170 or far back to Gangerolf or Harald Hårfagre. But they need to be known ancestors.
I think we all figure we're related back to King Olaf or
Yeah,
somebody like that.
yeah. In theory, you could use you could have used YDNA to actually prove if he existed or not. But to do that, we need at least 2 of his sons to have descendants today that we know for sure are his descendants. Mhm. Okay.
Yeah, this is really fascinating. It's a really valuable tool for digging back into those earlier centuries.
Yeah. Yeah, but it can, as you mentioned, Joel, I also had a criminal case where YDNA was used. And in that case, there was just one of these SDR markers like this one that was used to solve the case.
Wow.
Yeah, just one like this one. It might seem like a small marker, but that was what's used to solve it.
So you were able to show that the defendant connected back to the victim in this.
No, he definitely, the biological sample that was found on the girl was definitely from him. And the reason that we say it is because it was both a unique mutation like this, STR mutation, and what was also found was empty DNA or mitochondrial DNA that showed that the sample both shared his maternal lineage and his paternal lineage.
Okay.
Yeah. They also tested his brothers, and they also had a DNA sample from his father, and none of them had the same mutation. So my job was to map out all his relatives that shared his paternal lineage back to the 1700s.
Wow.
That was a lot of people, both in Norway and in America and Australia and everywhere around the world.
We all watch the programs, CSI and I mean, all these police programs that they collect something at a crime scene, and in about half an hour they have it analyzed and they know. I assume it's not quite that easy.
It's not like it's not that easy, but they tested around 40 people of his that shared his paternal lineage, and none of them shared the same mutation that he had. So we knew for sure that it could have been no other than him. But how the DNA was deposited, we don't know for sure.
Amazing.
Yeah, and that is also the same basics that we use in DNA for genealogy. we have some mutations that are actually unique, like in my own branch, this one that is unique for that sample, and probably the others that share the same lineage will also have this mutation.
Simon.
What? Yeah, Simon. Yeah, the earliest known ancestor is Simon here. Yeah. So this project is has a goal of mapping as far as possible all paternal lineages in Norway, and to find out what their connections are. So we have some parts of Norway that have been highly tested, like up in Møre og Romsdal. Don't know if any of you have ancestors from that area, but there we have a school project where they test around 10 students every year. We have a meeting where we pick out which lineages that should be tested every year. And then we can see also where these lineages in Møre og Romsdal comes from. So we'll see a lot of lineages in Møre og Romsdal. They have connection further south in Sogn og Fjordane, and we can see that there's a lot less connections further north, up to Trøndelag.
David,
And,
that
yeah.
information, was it used to make that map that you showed of Norway with the different purple colors?
Yeah, what
That
kind
was
of
the data that was used for that?
wait a little, I can show this.
Yeah.
Yeah, that is also part of our project, that we map which parts of Norway that has the highest frequency of each haplogroup. So this large one is from a project called Upedia. They have used DNA from public projects to calculate which areas in Europe which has the highest frequency of R1b. And then we have used the same approach in Norway, which shows that parts of Norway, almost half of all paternal lineages is connected to haplogroup R1b. And what is interesting for historians is that this can also prove how Norway was inhabited and how Europe was inhabited. So we know in the western parts of Norway, genetically, there's a lot of connections to the British Isles. So if you take most Norwegians who has ancestors from the western coast gets a lot of British in their ethnicity reports. And we have there's 3 major haplogroups of Norway. These are probably 3/4 of all Norwegians belongs to these 3 haplogroups. And this haplogroup is by far the most common. It's around 36% of all Norwegians.
Wow.
Yeah, and we see that in some parts of of Norway, around half of all the paternal lineages does belong to this haplogroup, I1, which is also my haplogroup.
Hmm.
And yeah, you can see that there's a high frequency around here. So this can be also used by those who are interested in finding out about how languages spread to Europe, and to confirm archaeological theories about how different technologies also were spread.
We have a third question from the chat.
Yeah.
room.
Yeah.
Someone wants to know about the isolated YDNA group that was off in Russia. And
Yeah.
what does that mean? He says, isolated matching group in East Russia / China matching Western Europe. Why?
Oh, okay.
It's
I'm
in
not-
the purple map, I think, or the red map.
Oh, this one.
That, yes.
Yeah, this one. Yeah. The theory is that R1b came into Europe from the east. So that's probably why you have a large grouping of R1bs here, because they spread in two different directions. But in addition to that, you also have a large cluster of R1b in the middle of Africa.
Hmm.
And we don't know why. Don't know. But,
Same hypothesis, it could have been the Silk Road trade route.
yeah. What is also one of the theories is that R1b was the group that brought the Indo-European language to Europe. So they were latecomers in Europe, and the I1 and I2 groups and the others, they probably had a different language than the Indo-European language.
Hmm.
Yeah.
Well, this has been really fascinating. I want to thank you for this presentation. Answered a lot of our questions, and I'm sure that people will have more questions. But perhaps we can invite you back again in the future sometime.
Yeah. Mhm.
Any
Thank you
last?
very much.
Thank you. Thank you very much.
Thank you.
Any last comments? Otherwise, I'll stop recording.